questioning that maybe it's not quite done regenerating, then it should stay in the dish for this week. Then the rest of them have to carry out the regular stuff for each week. So what we're trying to do is just limit the number of dishes that we have to go through. If your dish, if the notes have said there's nothing there or it looks like it's dead, then get rid of that dish and make a note on the paperwork that that dish is gone, because we don't want to keep changing water for something that really isn't going to make it. We've given it a chance, right? If it's fresh then, like you have one note that says, oh, I think it's done for, then go ahead and keep it for another week and we can try. But if it's disintegrated or if it's just plain gone, get rid of that dish. I know the white worms need at least another week regardless of whatever is going on with it, because we just cut them last week.
Do they have a weaker regenerative capability?
Who? Which one? The white ones?
Seems like they don't use as well.
I do expect them to go a little bit slower. Mm-hmm.
Their tail takes longer.
Huh?
I feel like their tail takes longer to grow back. Or
that could be. Yep.
Their heads are all formed enough.
Okay.
I look at them, but their tail.
That leads me to my next question, which I am thinking about, and it's up to you guys. I have your write-up for the class data right now we have as an entry in your notebook. Would you like to separate it out and make it a separate assignment worth 100 points?
Yeah.
The what?
The class data for the regeneration, yeah. So you'll end up typing the whole thing up and turning it in a full
We'll write
packet
up
to
on
me.
our own planarians, right? Or is it about the class?
No, the one for your own cuts, those are going to stay as a book entry.
Oh, yeah, those,
for the
right.
entire class data, because it has become such a big project and we're doing so well with it, we can separate that out and make it its own assignment and
like
make it worth more points.
Of like which one's good class. So
Yeah, yep.
The class.
So you guys would be looking at the regeneration rates between them, and if you want to add in the head versus the tail, which one you think is regenerating faster or slower, that's always a good discussion point. So you can think about those things too. So we'll expand it. It's making it a bigger assignment, but we're making it worth a lot more points, which is good. So we might need to earn a few more points. So we're all okay with that then? Okay.
But we're
Alright.
still gonna like follow the same kind of format with like hypothesis.
Yes, follow all of that stuff. It's just
Longer.
yeah, and we'll have to maybe try and find some examples of some good research papers, but so you should have a nice introduction, very good methods, the results with all of your data collated. However, I don't know how that's going, Shayna, if you started typing stuff up or not. Okay. That's all I need to know. It doesn't matter if you have or not. We're not done collecting data, so there's no reason to have that done yet.
Yeah, when's it good?
We're gonna make this due at the end of the semester still. It'll just be a big end of term project basically that you turn in. What else was I gonna say? I think that's all for that. Okay. Hello. We need to talk about what we're doing in lab today. This one. There's not a whole lot. Obviously your planarians take some time. And then we are going to be looking at annelids. So annelids are segmented worms, and we've got a number of representatives of each of them. So the famous one that belongs in this group is the earthworm, that one you're familiar with. And then Nereus is the marine polychaete that we always dissect for this class. So the difference is that Nereus, they have little kind of feet-looking things down sides. They have bristles, whereas the earthworms don't. So if you look at the clamworms, you can see all of the little frilly edges. So on Nereus, you're going to learn about those edges. You're going to be pulling off the little podia, putting that on a dissection scope, and learning the little features of that, as well as the internal anatomy of both earthworm and clamworm. Now, fresh earthworm bottle looks pretty. We do have some stuff that looks like this. Literally, it looks like just a bunch of those poor little snails got thrown into the graveyard. Okay, that's not what we want. So, this is a new jar of the Nereus as well, so if you want to use some scraps in there. You're going to open them up. You're doing some external and some internal anatomy for both. There's not a whole lot. I mean, they're pretty simple little animals. It's just that everything inside of them is kind of small. So when you give it a cut, give it a nice shallow cut and then just fold it open like a book and pin it down all the way down the body. You can put the earthworm and the clamworm right next to each other on the same tray if you want and compare them that way. Okay. There's also some cross sections of Nereus and lumbrigus that you'll want to look at. So those cross sections, they're out on these trays. I've lost some space to frogs today. They're gonna go away though, so we'll get our bench top back. So the slides are kind of all mushed in over here. So I'm having some lingering frogs. Okay. The leech, I still need to find the leech. He's in there somewhere or he's in the other room, but I just have very, very small leeches. In the past, I have ordered medicinal leeches so that the students can feed them.
Ew.
I leave that up to you. I only ever order them if you have people that volunteer to feed them.
Wait, they let them bite it? Are you serious?
Serious.
You have no
How do you
idea.
take it off?
How often do they
You let
Does it
them
hurt?
feed. They only need to feed like once a year.
Will someone in here volunteer?
Does it I'll do it.
All the boys should do
Would you
it.
really?
It leaves a scar.
Actually, when I have one on my ankle, I keep getting diabetes. So
Please don't order them. I
I was gonna see someone do it, but I'm not.
come back up Tuesday or Wednesday and someone's flying in the back.
How big are these?
People live in about this big.
How much blood do they suck?
They usually feed for half an hour and then they let go.
Are you serious?
So I don't feed them. That's not my thing. But I've had students do this in the past. So we put them on and let them attach. And I guess you can feel them at first. You can feel it more than you think you would, and then as they feed for a period of time, then you don't feel them as much. They inject an anticoagulant, so if you already have problems with, body issues, because this girl, bless her heart, last night, she's like, I'll do it. Like, all right, and she did, and she tells me after the fact, and she's like dripping blood everywhere.
Oh,
She's like, yeah, I usually don't scab over very quickly.
Oh, okay, not a problem.
Wait,
It's like
is it like-
information I'm gonna use. Or I'm gonna leech on you, but. So she literally, both of them bled for a good 24 hours, so it's a strong anticoagulant, so.
Don't take your fish oil before you.
Yeah.
Seriously.
They leave a scar that looks like the Mercedes-Benz symbol, which is why they wanted it.
Wow.
They have a scar?
That's
Yep.
the only reason they wanted to get bit by the leech?
They thought, I
No, you
don't know if it was the
don't.
only reason, but it was a nice side benefit that they were cool with.
Wow.
So I leave it up to you guys. If you have people that want to feed them, and you talk amongst yourselves when you decide you want to do that, I'll go ahead and order leeches for you. They come in a 2-pack, so I need people. And do not give in to peer pressure. If you have to do not want to feel like, wasn't it Rambo that went in one of those movies and he was in the swamps and he was covered in those leeches from him.
Does it hurt?
No, it was one of those kid
Does it hurt?
Yeah, Stand By Me.
Stand by me.
Yeah.
That had
I had one in
You can feel
there.
them when they first.
Do they like put something on you so you don't feel it? Like other
You
volunteers?
can, no, you can still feel it.
lidocaine or something.
No,
It's
some
not
of
like
them.
It's not like a
What's it
mosquito.
called?
You get
Oh,
lidocaine.
mosquito does that.
Yeah, it's not like that. You can still feel them. And you can feel them when they're like stuck and you can see that they're feeding.
Can you rip them off? Should you? I mean,
If you rip them off, panic. They are very hard to rip off. I did this with them last semester. I showed them I pulled on the leech,
Yeah.
and you could see the skin lift.
Ooh.
They make a very tight seal.
How do you get them off?
Usually you burn them off.
Either salt solution will take them off, but I don't like that one one because if you get salt in an open wound, it doesn't feel good, right?
No.
So we just let them feed until they're done.
Really?
And then when they drop off, or if you can get them to break the seal, like you can use your nails and kind of get underneath them.
I know, right?
Oh yeah, it'll come right back at you. It's always interesting trying to get the worm on the right person when I'm digging it out of the jar.
So
trying
you've
to get
been.
I'm Dr. Green. Lose weight now. Ask me how.
They haven't.
Yeah.
Blood.
Your blood?
Yeah.
Yeah.
You know, it's gonna be Halloween soon, so it's fun activity.
I think so.
You guys want to do it?
Wear a leech seat.
I want someone to
Steven, do it.
Yeah. Yeah.
Yeah.
Steven.
Whatever. So anyways, I'm gonna try and find our dead leeches. They're in a jar somewhere. We had a leech escape one year also. It died somewhere in this room.
You feel a pinch in your hand.
That's what I was thinking. Someone walked out and it's like stuck to his foot.
Yeah. Okay, nematodes. We have fun stuff for nematodes today. We've got living vinegar eels for you to look at. So the vinegar eels are here, so you're gonna just make a wet mount of them. They reek. They absolutely reek of vinegar because that's what they produce, is vinegar. So you'll smell them. What you want to look at for them when you do the free living ones, you want to see what locomotion they're capable of. Nematodes only have longitudinal muscles, they don't have any circular muscles. So they have a very whippy, kind of side to side locomotory behavior. That's what you're looking for. The prepared slides are all out, so we've got Trebatrix, which is the vinegar eel on a prepared slide. I only have one Trebatrix. I only have one of a lot of these, so they're kind of just on this one tray. The Enterobius, the adult and the eggs, and then Encyclostoma, Necator, Trichinella, and Ascaris. Trebatrix we have, yep. As far as I know, they're all here. And then you're going to do Ascaris now. Ascaris, I forgot to look at it. I didn't want to bother it all.
And you said for the locomotion on the vinegar eels, you want us to check on the locomotion?
Yes, please. Ascaris is here. Oh, here's my leash. There you go. Just trying to get to Ascaris. There's one. So I need to order more of those. Okay, so Ascaris, this worm lives in the intestines of pigs. So they're a very large endoparasite. They're a big worm. The difference between the male and female, the male has a hooky end. The only way externally that you can tell male from female. And even when you open them up, the gonads, they just look like little spirals of tiny, tiny spaghetti. We're just basically going to call it gonads. The only way you're going to know if it's testes or ovaries is if you've looked at the end of it to know if it's a male or a female. The intestine runs the entire length of the body, and it looks like a flat ribbon kind of next to the wall. So the curly stuff is going to be gonad, the flat thing is intestine, and that's about it. Because they're an endoparasite, so they don't have a whole lot going on. Alright, the onychophrans, I wish we had lectured on this stuff already, but Parapatus is just an adorable little worm. It's called the velvet worm. They get very big in the rainforest, and I'll show you a video of them when we get into lecture. But they have these cute little smushy legs. They look like little extendable cartoon legs. And they actually use internal body fluid to extend the legs. They're not like a millipede, which has an exoskeleton, and that's how they move their jointed appendages. These are non-jointed appendages. That's why they're here in this group, in the Onychophora. So they have glands on their head where they can spit out this kind of silk webbing stuff, and they use it when they're out hunting other animals for food. So they're predators, and they're very cool. And you get these little greenish blue ones. So you're going to put this on a dissecting scope, and you are going to be looking at the legs and at the anterior end. So you're going to be looking for the mouth and for the antenna, and I think for the gland openings. I don't remember if I told you to look for that. Oral papillae and mandibles. Yep, all that Then the cross section, we have 3 of these. And I remember from last year that one slide is definitely better than the rest. One slide has 3 cross sections on it instead of just one, so that gives you more detail.
That's the best one you see?
I like that one best. Alright, along with phylum Onychophora goes phylum Tardigrada, which is water bears. Water bears. Love me some water bears.
Tardigrades?
Yes. They, again, are going to be in a wet mount. Oh, their caps really tight up. They should have been loosened for air. Well, we'll see if they're alive. They may have suffocated. So anyways, make a wet mount. So they look like fat little bears, except they have 6 legs instead of 4. And they run around and they munch on things. So that's what you're looking for in them.
Put that stopping viscous liquid there.
They're usually not that interested in swimming around quickly at all. They just crawl on top of stuff. They have a piercing stylet, so what they do is they pierce into plant cells and they suck out all of the cytoplasm from the plant cells, and that's what they feed on. Okay, and then the predatory fungus is your last activity. So usually people do this lab to study the fungus. I do it to study the worm. So the fungus is here on these plates. We'll have to see how well they've grown. Oh, they're nice and furry. Ghosts.
Ghost of
The leech.
the dead leech. Yeah. The missing leech.
Ah. So these plates have been inoculated
Sucking.
with the fungus. So you can see they're kind of furry.
Yep.
Got it? Okay.
You have to put this in the dissecting microscope?
Yes, you're going to put this on a dissection scope, and you can see the parts of the fungus. You guys have had biodiversity, so I expect you remember a little bit of the anatomy of the fungus, that the mycelium is the main body. It lives down in the substrate or in the soil. The fruiting bodies, which would be either the sporangium or a mushroom if you see it, those are just for reproduction, so ours are not going to have that. The neat thing about this particular fungus is it will lasso the nematodes and prevent them from moving. They capture them and then they digest them. So that is what you're looking for.
So that's
today.
what they make, that's what they eat, that's the only thing they eat, nematodes or what?
They love to eat nematodes. And we haven't talked about them in lecture yet. Here, this is another one. Extreme close-up. You ready?
Wait.
If you look at the edges, you can see them squirming.
Gonna make you famous, doctor.
Wow, look at that.
I'm going to put a link to this.
Put it on YouTube.
Yep. To the people who
We're in the
control
back
my
row.
paycheck. So you can send that around. If you look, it's a liquid medium, and if you look at the wall, once you twist it around, you can see all the little worms squiggling on there.
Are there nematodes?
Those are some nematodes. So again, they're going to have the same look as the vinegar eels, but you're going to transfer some of them onto the fungus and then just watch them under the dissecting scope until you see that one gets caught and lassoed by the hyphae of the fungus. It may take them a while. Last year it took maybe 30 minutes before we had one that was for sure captured. And that culture is incredibly good. It's loaded with worms, so don't transfer too much. You're just going to take a little bit with your squeezy pipette and try and spread it out. Don't leave it as a big clump, or all you'll see is worms, not the fungus. And that'll wrap up your fun for today.
So the hyphae, that's the feeding, I don't know.
Those are the furry parts of the fungus that are extending up above the medium. The furry bit. Not this, I don't know why that chunk is in there, but yeah, it's kind of furry on top. Okay, so let me know if you have questions. I'm probably going to start grading
I
exams.
was skating at Limbo, and we thought that's like fireworm type of thing, that's what they call it. Is that anyone?
It was If, I don't know if it's a polycake, did it have
I
like?
have internet.
I think they say
Yeah, there are a lot of green
there's like, yeah, but it kind of like this thing, that's why I started,
We're going to check on the green, and I got
but it kind of looks like that.
anxious.
Yeah, it was like.
Hairs on it, like you couldn't even.
I just, you know, they had a marathon polycake that. Yeah, like, first. Like,
Yeah. I was spinning around. And
Yeah, I just got it, but,
Okay, so you need to run down, sometimes we run down biology for you.
will it bring me, you know,
Oh, you did? Yeah, I haven't seen any of the water.
I got, okay, the.
No, I didn't see
Mary
it.
said.
Oh. That's why you got so excited. You're like,
Yeah.
what?
I do it.
Yeah, once you walk in, they're more than likely. So the deadline's Friday.
This is
Wait,
good.
what do you? Yep, that's the first one. Yeah.
You don't have to do this another.
I have to go, yeah.
I'm gonna bring you a bucket.
Good life over.
And we're gonna,
Well, I'll sneak down and grab that paperwork.
yeah. I'll ask the secretary to tell you where to go. Yeah. She said that family and what else?
Oh, okay. What else do we need? There's another one too. No, sorry.
Well, a lot of people, yeah, they will start getting pigment on there.
I know Nick put an express on.
I'm gonna get them all in tomorrow. Sometime tomorrow.
I'm gonna get all the grades in tomorrow.
Do you need pins? You know, I have a bunch of pins online.
Hey, what is this registration thing, man? Is this to register a computer or something? No, I don't want to.
Yeah, I don't know.
I'm trying to get into Blackboard. Not letting me.
And I forgot the pen. It's weird. That's why. I was like, I know there's something missing, but because it's in I was not thinking it was a struggle over there. I was like,
The central communication.
oh, you don't even want to know what I call it. I call it Umbra. I couldn't get enough. That's why I
I
call
want to
it
get into Blackboard.
That's not brand. I know.
do something for myself.
No, that's
It's
not
not
what
a
I
brand.
meant.
Yes, it is.
Or like,
No, it's
used
not.
to be.
I think
Something.
so. I don't
Something
know.
like that.
That's probably why it was in my head or something.
Right there.
This is for minus commuter.
Yeah, and then once you get there, go to Blackboard.
That's what I did. See, I'm already in
Yeah,
here.
should we or should we?
And then go down.
Sure, on everything fast.
Or no, you go to students, right? Oh, you are.
Yeah.
Down, and it's over there somewhere on the left.
That's where I went. I hear Blackboard. I clicked on it.
Okay, tell me what to do. Alrighty then, following the
It's taking me here again.
following
Now you gotta put.
may be done.
Oh yeah, I don't know what's going on.
One that has
That's a different page.
either case the worm should be kept submerged under the submerged. It says submerged. That's I just missed my part because I cannot see. Should be kept submerged. In general, basically, do not work well on freshwater. Very much.
Okay.
Where the rest of the earthworm is supposed to have now for the following information concerning the live learning. The same specimen of Mary's and examine both surfaces. There is a slight mid-ventral depression that runs the length of the worm. Anterior posterior.
and children get so stressed out.
I know, I'm like,
Considering
sorry, I'm not my name on it.
I want to be done, but it was like, what are you doing now?
Is that okay? So upstairs. Ours? Okay. Okay, examine the anterior view and look at the proscenium.
Isn't that like
Eyes, palate, and antennae. Yeah, but we don't know the proscenium.
You guys are doing the work?
What? Yeah, we're doing the worm. Look, proscenium. But you can't come in. No, he's not allowed to come. Let me find my microphone. Wait, get in that section.
who's got my scope?
Uh-huh. Nick, I'm sorry, this is mine. I'm repossessing it. Sorry about that.
Hey, don't ever do that, man. Don't
Shut
let
up.
the- Let the woman control you, man.
He knows, no, he's a gentleman.
Oh.
So you should learn from him.
Oh.
Yeah.
You know me so well.
It's the worst. Yeah. The worst ever. Yeah, I know. Okay, proscenium, I want to show you. Does yours have that? What? Proscenium? It's like this little weird part. It's on top of the head.
Oh, it's like the head of the back.
Oh, wait, I don't know. Anterior view, it says.
Oh, it does?
Yeah.
Mm-hmm.
Look right here, Jan, so you can see.
Okay. We'll be able to see that. I can see it already. No, go away. Oh, it's a good job.
But one time you actually want to use the back leg. Yeah, but here, I heard about one. Marvin, we're just kidding. No, we're not. I actually love it. Oh.
Yeah, because I set it down this way, and I was like, oh, it's gonna
Oh, so here's the thing. You can be mean to people. People can be mean to you.
Yeah, I got it.
Sure, bring it.
Oh, who's this? This yours?
each one is called a separate.
Pinned her right on the head. What? See, you, Nathan likes you a lot. No, it's not that. It's just this part. I don't know if this is the palate.
But it sucks when
Should I bring the recycling scope over there?
Now you get,
Yeah, well, we don't know
now you
the
get to find it the
actual
physical of you.
Well, that comes out, maybe. You know. Maybe. The pharynx extended, the pharynx retracted.
I guess I'm
See,
just
this is
gonna
right here.
hover over them and see what they're doing.
I know, it's weird. Comes out.
Here, Liz, it's here.
I bet that works like another thing for your gel filled or.
It's the
you can't
smallest
grab 'em.
antenna. I don't know, it's weird. It's like a small antenna. It's smaller than the larger
Oh,
antennas.
it's here then. Actually, I think
You want to look on this first? Did you already see what it's
Love that tripod for that.
Me?
it comes
No, I'm talking to Nick.
Yeah,
This.
man.
That
Kodak?
Yep.
When you
Oh,
on
trays.
another second, would you mind coming and looking to see if we're looking at the right thing?
Sure.
Take one of these.
Where am I? Oh, there you go.
Oh.
Video, please, Rick. Not yet. What are you looking at? We're trying to find this prostomium. Okay. So that is part that doesn't always come out. So the peristomium means around, and the prostomium is the entire part.
Yeah.
Okay.
Go to that section too.
Because this is the palp, and it's covering most of the prostomium cut off. From that view, see how they're kind of off of the prostomium.
You don't have to dissect nearly, right?
The prostomium is the front that has
Oh, yeah,
all
we
the
have to see
antennae.
your film.
I think it starts with
Peristomium
external
is that
features,
first
then
segment
go to-
after it, which doesn't have any parapodia off.
But it has these other, oh, it doesn't even have any of these.
One of those stress dolls, it's like
Okay.
plastic with a.
Alright, so it's just the top.
Yeah, kind of slowing it down.
So they kind of, this whole thing
This whole
popping
thing
out of
comes
that.
out. Yep.
Holy.
First we have to see them dissect them.
Anything.
And open it, you'll see the little
Damn,
dot
Sam.
there.
Not kidding around.
So they,
You're gonna need pins to
Predatory worms. They're very cool.
I saw this cool predatory thing.
We have to see them dissect the mic. We have to see external features. And then the internal.
Okay,
Like
eyes.
hornets that would attack honeybees.
Which is this one?
Maybe 10 or 20 of them would mob a colony and kill the entire
The eyes are
colony.
tiny.
Yeah.
Yeah.
We have to somehow.
don't drop no juice on my camera.
Holy shit. Things are big.
Oh, you got it?
I can be clumsy at times and then drip all over his stuff.
They figured out they couldn't figure out why these things took the dead.
They're two black dogs.
They're eliminating
They have
this.
4 eyes. Where do you see that? 4 eyes, that's me. Oh, cool.
Little guy isn't always.
There's too much faith behind it.
No, they really do have some ways that they can handle it. Find that. This one's losing its skin. That's why. But I got they're awesome. That little face on the dissecting scope there. The palp, yeah.
Is the palp part of the prostomium? Is it like
The palps are off of the prostomium. Mm-hmm.
Karen,
Yes.
will you be on campus on Friday?
I am. I teach. Fridays are a goofy day for me. I teach 2 hours over at Raleigh. From 9 to 11 and then from 12 to 3. So I can see you in between them if you're available. I just won't be in this office. I'll be over there. Al's fine. Yeah.
I'm coming into the
Worms? Okay. You have to be to work at 11. I need to see you before 9.
Really, I guess I can today.
Okay.
Anything? I'm not sure
You're
yet.
not sure at all?
Yeah.
Okay.
I'm hopeful, but
I'm hopeful as well. Okay, so,
Yeah, to see the jaws.
but you
if
will have,
had
you
us-
will
yeah.
have results before.
I'm gonna do them, I'm gonna try and start grading while you guys are actually working today. And so that I can get them entered in, tomorrow. Hopefully,
It is. We have.
yeah,
it's
yes.
right now.
Yeah, Friday is the day, so I mean, at the end of day Friday, then it's done.
Okay.
So I'll put you down, what, like 8:30 or something maybe?
Yeah.
Okay. And you should be in a comment if it changes, but.
Okay.
Okay, just to double check again, I know I always ask the same his little like things, eyes, and periostrobes, that like shape of thing right there.
Very good. No.
Prostomia.
The peri is the next, this
The next segment. This right here.
Yes, because it doesn't have anything.
Very great. Chemical reception, okay.
Yeah, that's fine.
Dude, you're so obvious. So obvious. Well, I feel my hair being pulled. I didn't Nathan gets all quiet. Nathan's quiet for 2 seconds for me to breathe. Unless he's up to something. Yeah.
How do we see the jaws? We have to like pull it
Yeah,
out.
cut them open.
Oh, cut it.
Yeah. You can end up cutting down through the peristomium, through the prostomium as well. To get that. And it's going to be kind of lodged down in the neck.
Okay. Alright, so the periostome is the whole thing about.
What's the peristomal series? How? Alright, let's cut it.
Okay. On the dorsal side.
Wait, so first, I guess we have to see the.
Yeah, these are the things that you This is
Yeah, that's the anterior.
Oh, I
And the posterior side doesn't have. Oh, but the siri are on each side though. You are the caudal siri. And then you have on the front. You see the ones on front? On the anterior? So to see the prostonium in all that? To see the prostonium, I have to open it up first.
Yeah.
No, not-
Prostomium, no.
This is narrative, right?
Prostomium. Flip them over actually, where you can see. Then you should be able to see the palps and the antennae, and then you know you're on the prostomium.
You're gonna use it, is it? You're gonna have to start cutting it out.
Because I think I could hydroid. That's close.
That one's probably not. You got something there. Maybe, yeah.
I think I put
Oh, the actual organ, yeah.
or hydroid, can't remember. If I put hydro, okay.
No, because it was over here. Yes. And hydro was that free living one.
I think I put hydroid.
those are black spots on this anterior.
40?
The what?
All those little black spots on the anterior.
Here, go. You can bring it here.
What's that?
You can bring it here to open it up. Oh, no. The eyes are those black spots. The prostonium is where the eye is.
There's no those things.
You see?
So map
Because I see here the eye. If you look at the
There's the mosquito.
oh no, this is the intern. If you go to the dorsal side, so kind of trying to see. Like, what are you looking for?
I'm just looking at there's a bunch of little around the mouth there, right? Is that from the little
Review, I guess.
should probably wait over there. Take some of this video.
But the, like everything like inside though. Like for
The
example,
jaws are gonna be done inside.
like
Is that what you're working on?
Like
That
a-
you're gonna have to cut open.
I can't see the prostonium.
What are the little black spots on?
Oh, that's the
Yeah,
prostonium.
see these little things sticking out? Here, you've got antennae, and these two things that kind of look like little legs with feet on them, those are the palps.
Okay.
So that is off of the prostonium. So if we look off this end, look how fat he is right there. That's a good one. Yep. So prostonium is here. We gotta move the tentacles out of the way to see the eyes, because the eyes are gonna be about right there. These are the palps. And then this is the peristomium here, this first segment that doesn't have any parapodia off of it.
And again, the prostonium is which part?
This. Between the two palps.
Oh,
Between
okay.
these.
Oh, okay.
Then move this over. And see the eyes.
Dorsal longitudinal muscles. Wait.
See the little black dots? Those are the eyes.
And they're actually just called eyes.
Circular muscle.
They're predators, so they're actually very well set up for sensory stuff.
So I actually see more than this.
I think for me. Is Neris's oblique muscle like the circular? Is that a different?
Different.
Hey,
That
Steve,
one actually,
wanna take a
instead of
picture?
being completely
Yeah.
dorsoventral where it goes straight from the back to the belly, it's at an angle. So it comes like from the side to the ventral.
That's the oblique.
Yep.
And then the
Pretty
circular
nasty
one.
looking things under the mouth.
Circular.
It's kind of whatever it is.
Yeah, that is separate. That oblique is probably not gonna be.
It's not as bad.
Beautiful.
Yeah. Two black hooks, like Sam. Oh yeah, they're big. But there's one there and there's one here. I see that right there. We could be It's true. This was, yeah. Here, wait, I'll show you.
So we have to open it up to see the jaws.
Yes. Yep. Sorry.
Sorry, I'm thinking a little bit.
Not this part, they're in here. This one is so high the top.
You ever seen an antelope?
No, it's on over there.
You ready, Steve?
Yeah.
I would just like this one.
You want another microscope?
Just can't keep it still.
Is this one just a hit?
Yeah, you get that one, but push it down a little bit first to focus it. Remember to get the bra on the bottom too.
See, I told you that.
And parapodia.
Now I
We can move up from the tail. Doesn't have to be.
And from anywhere on the body. Every single little structure around the outside is parapodia. Where are the
Good. Okay.
roundworms and the cornmeal that has two.
Oh, yeah, I just saw them. Nathan?
Man, these are nasty flies, man.
They look like fireworms.
Makes me like.
It's that big with the black cat. Oh, I see them. Thank you. Alright, what's next one? We want this thing up there. Looks like it has a beak like that.
Where's the little podium? So what's the notopodium of? Dr. Green?
Yes.
The notopodium? It's not showing it. Oh, I see it now.
Got it?
Oh, it's within the parapodium.
Yeah. We need what?
So to read the internal structure, we just need the interior end, right? So I can like cut it in the middle and.
You want to try and do, you want to open them like a zipper.
But I'm saying to look at the parapodium.
Just take your forceps and literally just pull one off.
Of the nearest.
The whole thing should come out.
Alright, what else is the nearest camera? We need septum. Wait, you can find the seed? No. She said something about pigtail. Maybe she noticed the difference between these two. Okay, and then we need to do internal, okay. Septum, dorsal fundus, here, look, right here. Okay, wait, esophagus, septum.
Oh, this is some small stuff, man.
Want me to go to the cut open, right, so you can see everything?
I think we need more, so he's pretty much done.
There's nothing left of that.
Is that the POs?
Like a 1, 2, 3, 4, and that.
So Miss Green?
That's a perfect.
Yank one?
Yeah, I did. But I don't know if you can see really well
Put it on the
nor-
slide, it'll be easier to work with.
Put it on the slide, right?
Yeah.
Stop, Nathan! I'm gonna slap you. You spread the stench when you do that. Look, yeah, you're going like this. Esophagus, gut. Is the gut the esophageal cecum? No. But it looks like it.
We're doing the second part to remove a parapodia and put it on the slide.
You have pins, the small pins.
Is that the
Oh, for structure? Yes, please. Okay.
What is a neuropodium? What is a neuropodium?
Here's on this side here. When we take out the parapodium, which is just like this, you just grab this and walk it up. Yeah, the foot, like the foot. And then on the foot, you can see the lotus is tied and stuff. I got it on the slide now. I don't know if you want to look it on the slide while I start cutting the head. Or just slicing it up. If you want to look I got, you want to look at I got my gloves on. If you want to start looking at it on the slide. Let's start up with Do you have another?
You guys should have your handles for me dissection kit.
Nope.
I have
Nope.
blades with it, but I don't have handles.
Yeah, I'm thinking I got like 16 wrong, which is not really passing. and it just kills me when I get one wrong that I knew was.
Yeah, I've been thinking about that, about the proglottids. Couple of questions, I'm like, damn it, man.
Yeah. I don't know. I'm a bad test taker. I read the question wrong.
I just go blank when I'm there.
Yeah. I have trouble remembering spelling and.
Does anybody in this classroom own a Honda Civic?
No.
Okay. Why did one just get stolen?
Or towed.
No, they said that there are a number of auto thefts going on involving Honda Civics, but I don't know if they're break-ins or.
That is too, so a whole different other ballgame, so that works.
Well, if they wanna break in, they can handle my car.
Anyways, I don't have a Honda Civic either.
I have one in Chile.
So it's safer than your baby.
Oh, wait, leave it like that, cause I think we get it. Alright.
Are the pins for holding out the specimen somewhere?
Hard to see it.
Yeah,
like-
either the third or the fourth
Alright,
side.
look at these things right here.
Unless somebody picks them up.
I think these lines right here. Yeah, so I think all those little things, those lines. Alright. Stink at that. Really? Alright, yeah, I can't.
Now we know.
I don't want to overstate things at all, Dr. Green, but Bruce from the microscope company,
Yep.
what a guy.
Yeah.
He gave me $1,500 worth of equipment without, not, he said, take it home, try it, make sure, it's what you want. And, he gave me the Olympus CH, like we use, CH2, and then also another one, I don't know the name, but it's a dissecting scope, but it's a $1,000 dissecting scope for $750, and it's slightly used. But he gave it to me like on a handshake kind of a thing, and he's going to be here not this Monday but the following Monday, and I'll decide and pay or whatever then. I told him about you, and I think he's got an inventory what you might be looking for. So he may call you, or you may want to call him. Yep, really nice guy.
Stephen, where did you see the box of pins? Anything smaller than
Over there,
that box right now.
on the last bench.
What part was this on the slide again? Or just the whole-
This is the
head area or one of the leg area?
I just took it whatever from whatever, and it should be there. So this picture.
That's what's on the slide?
Uh-huh.
Could you pass my book, please?
I don't know if you want me to do that or not. It might never smell the same.
Did we cut him on the right side though?
It looks like because they're really thin, kind of. Because it should be on the fan.
Do you have like a plastic thing for this? That'd be perfect. Like a plastic model that you use for this. Oh, yeah.
Did you guys find everything in it? Oh, that's the other one.
Yeah, I just got mine open.
Have you guys found it?
Found everything.
Oh, yeah.
We're done with that.
Everything?
Everything. You're not gonna put the leech on?
What leech?
The leech. We're gonna get leeches.
Yeah, of course.
Will you do it? You're a baby.
Yeah.
Nick, will you do it? No. Baby. Steve?
I'm with that.
Nathan, will you do the leech if we get leeches?
You know, I really shouldn't.
Should?
No, I really shouldn't with my skin stuff. I wouldn't mind, except I don't know. It might be somewhere I'd end up bleeding for a while.
Compression, man. Compression. It'll stop bleeding. You won't bleed to death. You won't bleed out. I can't believe you guys. But you would do it.
Yeah, me? What? I'm so manly to.
You're so manly.
I look so
But-
strong
but
to
clearly,
put a
yeah.
leech on me.
Clearly you're not.
Yeah, I'm to disappoint you.
No, it's okay. Oh, that's the
If you do
earthworm?
one, I'll do it.
Yeah.
No way, man.
No way.
Dude, I won't even touch a frog. I'm gonna put a fucking leech on my arm. But that one has, like, the stuff's clearer inside of it.
As earthworms go.
Huh?
As earthworms go. It's pretty clear. I feel like when you enter like some sort of competition and you like lose, you know what I mean? That kind of feeling, like, man.
How about a test?
Yeah.
I don't know, it's saying here that she's gonna agree with mine. It's gonna be like a total ass.
You know, I don't know how to convey this, but it's like, if you were to leave the answer, if you were to leave the question blank, all right, yeah, you deserve, you don't get a point. You give an educated, guess that is reasonably, thought out. All right, a little something, something. Maybe not a full point. Maybe not even a half a point. Maybe a quarter point. But if you're trying to like nitpick on going from a B to an A, yeah, if I'm a teacher, I don't want to hear that. But if you're trying to like, fail or not fail, all right, let's work something out. I bet you didn't think this was going to happen to you today when you woke up. You didn't wake up.
Yeah, I hadn't been awake for a while.
Yeah.
One of those specimens, I forgot the name, was from 1968. Yeah, in a jar. Like, I'm not opening that. I can imagine how bad that smells.
It gotta just be mush.
Yeah. No, it wasn't one of these. It was a
Structure to it?
yeah. It was a nice, clean, clear specimen in a clear jar.
How long will something like this last in a jar before it just turns to mush and really?
Good 10 years or more.
Wow.
Which is why some of the specimens we have look way.
Yeah, because they're 15 years old.
Yeah.
Reagan was in office when you were caught, dude.
Like, when you go to some museums will have the babies in the jars.
Yeah.
I wonder how long will something like that last? I just wouldn't have to change those out.
I don't know. I mean, I know it's long term. I don't know if it's indefinite, but
They're preserved for the long haul. They really are.
So they'll last a while?
So, that's the green.
The museum in Chicago we always go to. That's a good display.
yeah.
So these are the septa.
Yep.
These are septa.
Little line in
Which one
between
are
each one.
these? What's the dorsal blood vessel? The thick one?
Those are those.
Yeah, I'm putting
See
it
it on
here.
the other one.
I am not 100% sure. I just took him. I'm not sure what he is exactly.
What's that?
I just took him. I'm not sure what he
It
is
might be-
exactly. If it's
usually-
earthworm.
Oh yeah, it's the other one.
And it's probably up in the top, so.
What'd you call
If
it?
you just make
It's-
drawing
it's
about
it's
where
the
it should
second
be.
one.
Second.
Yeah, it's, I believe
Yeah,
it
you can like
is.
This one?
that's fine.
And what's this? This is the
So that's
pharynx.
part of the pharynx, yep, and then that's gonna
Oligochaeta.
continue on into the.
I believe so.
And where's the gut though?
The gut runs the entire length of the body then.
I really
This
don't
is
know.
I don't know
two
either.
that runs all the way down.
All I know
Well,
is
it's
it's
not
a
even nice one.
big one. Yeah. Smell each.
No, because the prostomium is
Here we
the
go.
first top of the dot. Okay, that's
Lumbricus.
Lympicus. Okay, that's this one. Yeah, you're right. It is. It is
That's the first one.
the second one.
Yeah.
All right. So write a definition and function of the following terms. Clitellum. So we do the specimens. Label the anterior, posterior mouth. Prostomium, peristomium. Clitellum. Male genital pores, oviducts. Let's see what this is.
That's all it is. Dominant region. So it's a dominant region of the epidermis in sexually mature individuals. It's a band-like glandular structure, maybe one or many cells thick. Varies depending on species and time. Produces mucus during copulation. Secretes a cocoon.
Sounds fun. Alright, so we have the longitudinal muscles, cervical muscle, cuticle, epidermis, seta, parasitic nematode, nephridium, schizosomal.
Alright, if you guys wanna come look at this, I started some worms on a plate of fungus. I had it on a regular compound microscope instead of the dissecting scope, so you can see it a little bit better. So this is what you'll need to watch. If all of the worms are, and do these on your own plates too, because they don't all work. You want to watch the worms that kind of get away from everybody else, and they venture out into the fungus farther than the rest. The circles that are in there that they're swimming around, those are pieces of potato medium that they're feeding on, so just ignore those.
Nasty.
Crazy, huh?
Yep.
Nematodes are wild.
Look at it over there. It almost just looks like I could just build some sawdowns. Carbonation's foaming up a little bit and bubbling around.
Yeah. Looks like applesauce. It's alive.